Some molecules are capable of being excited via absorption of light energy to a higher energy state, also called an excited state. The energy of the excited state, which cannot be sustained for long, decays or decreases, resulting in the emission of light energy. This process is called fluorescence. To fluoresce means to emit light via this process. A fluorophore is a molecule that is capable of fluorescing. In its ground state, the fluorophore molecule is in a relatively low-energy, stable configuration, and it does not fluoresce. When light from an external source hits a fluorophore molecule, the molecule can absorb the light energy. If the energy absorbed is sufficient, the molecule reaches a higher energy state called an excited state. This process is known as excitation. There are multiple excited states or energy levels that the fluorophore can attain, depending on the wavelength and energy of the external light source. Since the fluorophore is unstable at high energy configurations, it eventually adopts the lowest energy excited state, which is semi-stable. The length of time that the fluorophore is in excited states is called the excited lifetime, and it lasts for a very short time, ranging from 10 to the minus 15 to 10 to the minus 9 seconds. Next, the fluorophore rearranges from the semi-stable excited state back to the ground state, and the excess energy is released and emitted as light. The emitted light is of lower energy, and thus longer wavelength than the absorbed light. This means that the color of the light that is emitted is different from the color of the light that has been absorbed. Emission of light returns the fluorophore to its ground state. The fluorophore can absorb light energy again and go through the entire process repeatedly. The cyclical fluorescence process can be summarized as 1. Excitation of a fluorophore through the absorption of light energy. 2. A transient excited lifetime with some loss of energy. And 3. Return of the fluorophore to its ground state, accompanied by the emission of light. The light energy emitted is always of a longer wavelength than the light energy absorbed, due to the energy lost during the transient excited lifetime, as shown in Step 2. A fluorophore can repeatedly undergo the fluorescence process, in theory, indefinitely. This is extremely useful because it means that one fluorophore molecule can generate a signal multiple times. This property makes fluorescence a very sensitive technique for visualizing microscopic samples. In fluorescence, the visible spectrum is composed of light with wavelengths ranging from approximately 400 nanometers to 700 nanometers. Light waves with shorter wavelengths have higher frequency and higher energy. Light waves with longer wavelengths have lower frequency and lower energy. As we stated before, an excited fluorophore emits lower energy light than the light it absorbed. Therefore, there's always a shift along the spectrum between the color of the light absorbed by the fluorophore during excitation and the color emitted.
Instrument is used to study fluorescence emitted by an analyte. The principle of spectrophlorimeter is it uses the fluorescence produced by the analyte to perform the qualitative and quantitative analysis on it. The fluorescence emitted by a particular analyte has a specific wavelength associated with it, and the amount of emissions gives one the quantitative measure of the analyte. Let us start with the construction. The first component of the spectrophlorimeter is the light source. The lamp or the light sources that can be used include xenon lamps, high pressure mercury vapor lamps, lasers, or even LEDs. The laser and LEDs have a very specific wavelength. The next component in the spectrophlorimeter is the exciter monochromator. This monochromator is placed between the light source and the cuvette which holds the sample. The exciter monochromator can be a prism monochromator or a diffraction grating. The sample under test is placed in the sample holder. A transmission detector like a PMT measures the transmitted light from the sample. The next component is the analyzer monochromator. It is placed perpendicular to the incident light beam. The analyzer monochromator is also a prism monochromator or a diffraction grating. The next component is the fluorescence detector, which is generally a PMT. PMT is used as even slightest radiations or low intensity radiations are easily measured by them. Coming to working, we can see that the light from the light source passes through an exciter monochromator. Here, only the required wavelength of light passes through the exit slit. This incident light is absorbed by the sample or the analyte. The remaining light gets transmitted and falls on the transmission detector. The sample gets excited by the incident radiation. The phenomena of fluorescence takes place, and fluorescence of a particular wavelength is emitted by the sample. This emitted radiations is made to pass through analyzer monochromator, which filters out any stray light mixed with fluorescent radiations. Once the fluorescence exits the analyzer monochromator, its intensity is measured by the detector, which is generally a PMT. The wavelength of fluorescence is very specific and depends upon the molecules or electrons present in the sample. Thus, one can identify the compounds present in the analyte by studying the fluorescence wavelength. The intensity of fluorescence is dependent on the number of a particular molecule or electrons being present in the sample. Thus, intensity helps in performing quantitative analysis of the sample. This was the working of the spectrophotometer. Now, the advantage of using a spectrofluorimeter is greater sensitivity is achievable since background signal is zero when compared to absorption measurements. Differences in wavelengths between incident and emitted light helps to discriminate between substances having similar absorption characteristics.
Hello everyone, now we're gonna have the interview about fluorescence spectroscopy. So I'm gonna interview now, and for a start, what is fluorescence spectroscopy?
Well, fluorescence spectroscopy is a type of electromagnetic spectroscopy as any other type of spectroscopy machine, SAS and all other, but it's used to analyze the fluorescence in a sample. That's the main definition. It's from the word fluorescence.
Oh, that's why the word fluorescence came from it.
Yes.
Nice. Is it like any difference from other, like in the aspect of like the machine itself, the components of the machine? Is there any difference from other, or is like that have the same thing? What are basically the components of the
Basic as same all of the other machine. It have a source, sample,
Okay.
then monochromator. Lastly will be the detector and the machine computer and ID device. But it will be different, of course. There's the source. The source they use UV light.
UV light in the source.
Yes, they use UV light, beam of light, which is ultraviolet, same as the ultraviolet in UV. This will lead to the excitation of electron from ground state to excited state.
I see.
Same principle as the UV. And also it have absorption. We can measure the wavelengths by absorption, emission.
Yeah. Like emission and absorption
Yes,
spectroscopy.
like other spectroscopy. And as for sample, there will be usually a sample, and it's used to know the intensity fluctuation from the emitted light measured. So we have sample, they measure the emission light of fluctuated, and also then we have monochromator. It's used to filter the wavelengths, and the specific wavelengths it will detect.
Like any function of any monochromator in any machine.
And at last we have the detector that detect, and we have computer. We
That
see
give us the data.
The
I see. So what's the use of this like machine or this technique of spectroscopy? So what are the application basically of fluorescence spectroscopy?
Well, fluorescence spectroscopy, its application generally, it's used for biomedical
Biomedical applications.
Yes, and it's to use mostly to analyze organic compounds.
Organic compounds.
If any organic compound contain fluorescent or any other compound can be sent to analysis. And also, it can use for skin tumors.
Skin tumors.
Yeah, as a start.
Interesting.
When it first started, it can be used. There are atomic fluorescence spectroscopy, AFS. It's a technique also useful for analysis and measurement of compounds that are present in air and water, like water analysis. Also used in environmental study.
Environmental studies.
Study water.
To test the
Because
water.
it can use it mostly fluorescence used to detect heavy metals.
Yeah, that's why we have that's why, yeah, like mercury. I reckon that we have another kind of spectroscopy that tests like the light metals. So this one only for heavy metals. I see.
Yes.
So you are doing FYB right now, right? Have you encountered with this machine at all or nothing?
No, but I'm doing also organic synthesis, but mostly as general other techniques. Like
Other techniques.
GCMS.
But in
because it, like I said before, it's used for heavy metal.
For
My
heavy
work is not
metals.
heavy metal, like mercury.
I see.
And other
Heavy metals.
metals. Yes, and it's usually used. Fluorescent detector are used with HPLC.
So we encountered this machine like when we use it as our like in our lab as a lab report or as a lab technician when they showed us how we do it. Do you see any difference between it and other instruments? Like
Yes, each one will be of course different. For example, for data analysis, when you have low concentration of fluorescence intensity, this will be proportional to the concentration of the fluorophore.
Fluorophore.
Yeah, they are proportional. Not like UV-vis spectroscopy.
UV-vis.
There's a standard device. It's independent of spectra and not really easily attained or brought. And there's also many factors that affect and distort the spectra, like any other. And of course, in this machine, there will be corrected diagram and everything, but mostly be proportional to the concentrations of fluorescence.
So in the fluorescence spectroscopy, there are like also errors and something. When we use the machine itself, like, do you see it easier? Like, is it easy to handle than other machine, or it's harder to handle than other machine themselves?
All machines, especially also fluorescent, it's very sensitive.
Very sensitive, right?
And expensive. So anything wrong, it will lead to expense, and that can be all afforded.
So thank you very much for this interview.
Welcome.
Thanks.
Fluorescent dyes, fluorescent stains, or fluorescently tagged antibodies to detect proteins, structures, and/or processes within cells, tissues, and organisms. Using microscopy or flow cytometry experimental techniques, we can detect the presence, localization, and amount of fluorescent molecules. Fluorescence is frequently used in such experiments because it can be detected in a very sensitive manner, leading to quantitative results. Some examples of fluorescent molecules are DAPI, used to detect DNA, ethidium bromide, used to intercalate DNA in agarose gels, and green fluorescent protein, a naturally occurring fluorescent protein found in jellyfish.